Welcome to People Behind the Science. I'm your host, Dr. Marie McNeely, and I'd like to thank you for joining me today for episode 699 with our guest scientist, Dr. Daniel Czyż. I hope you enjoy getting a behind-the-scenes look into the life and experiences of another researcher in today's episode. And if you want to get to meet even more of today's scientists, listeners, you can check out our website at peoplebehindthescience.com or subscribe to our show on your favorite podcast platform. Today, Daniel has some exciting stories to share, so get ready to meet another one of our remarkable people behind the science after a short message from this episode's sponsor.
We are research.
We are discovery.
We are scientists.
We are diagnostics.
We are Lampire.
For more information, call 215-795-2838 or email lampire@lampire.com.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Listeners, today I am excited to be speaking with our guest scientist, Dr. Daniel Czyż. So Daniel, welcome to our show today. How are you?
I'm great. Thank you very much for having me here, Marie.
We are thrilled to have you with us, and I'm excited to learn more about both you and the wonderful work you're doing. But let me first start by telling our listeners a little bit more about you and how you got to where you are today. So listeners, Daniel is an assistant professor of microbiology and cell science at the University of Florida. He completed his undergraduate training in biochemistry at the University of Illinois at Chicago, and he was awarded his PhD in integrated biological sciences from Northwestern University. During his PhD, Daniel spent 2 years as a visiting scientist at Argonne National Laboratory, and afterwards he conducted postdoctoral research at the University of Chicago and the Howard Taylor Ricketts Lab at the Argonne National Lab before joining the faculty at the University of Florida, where he is today. And Daniel, today we want to get to know you both as a scientist, of course, but also as a person. So can you start by telling us what you like to do when you're not doing science?
When I'm not doing science, I am a regular family guy. Mm-hmm. I have 3 daughters at home and my wife, so we usually enjoy outdoor activities. We live in Florida, so we're very close to different beaches. We're 45 minutes away from the Gulf, 1 hour, 45 minutes away from the Atlantic. So whenever we had any time off and the weather permits, we go to the beach. We enjoy camping, fishing, and we love traveling around as well. COVID kind of slowed us down with the traveling plans, but we're starting to pick that up again slowly.
Absolutely. Well, it sounds like you've got a lot of wonderful activities keeping you and your family busy when you're not very busy in the laboratory. And you were doing some amazing work in the lab, so I would love to talk about that as well. How do you describe what you do to someone who is not in science or maybe isn't particularly knowledgeable about your specific field?
It's also somewhat challenging, right, to explain. And my mom always asks me, so what do you work on again? And I have to put it in a very plain language. So my lab works on 2 different research areas. Half of the lab is trying to develop new treatments to kill antibiotic-resistant bacteria. So we're pretty much hitmen that are trying to develop not new antibiotics. Antibiotics target bacteria directly, and eventually bacteria develop resistance to all sorts of treatment that targets them directly. But we're trying to explore something non-traditional. So we're using bacteriophages, which are viruses that specifically attack and kill bacteria. We're using silver nanoparticles. These are spheres of silver that directly can target these microbes, and then they can also potentiate the activity of specific antibiotics. So we could enhance the potency of already existing antibiotics that are losing effectiveness. And we're also working on host-targeted approaches where we're trying to enhance the ability of our own immune system to fight off bacteria. So there's a subset of cells in our body called phagocytic macrophages, and these macrophages, they find and engulf bacteria. So what we do, we find small molecules or small chemical entities that can target these specific cells and can enhance the ability of these cells to either uptake bacteria, or we could inhibit the ability of these macrophages to uptake bacteria. So you could imagine that this could work for 2 different indications. we could enhance the uptake of common bacteria that live outside of cells, or we could inhibit the uptake of bacteria that require to enter cells. And these could be Salmonella, Coxiella, Rickettsia, Brucella, Neisseria, and all these other they're called intracellular bacterial pathogens. Now, the other half of my lab, I know that's a lot already, but we have other research projects that we're working on. The other half of my lab is trying to understand the effect that bacteria in our gut have on our brain. So we're studying the effect of bacteria on neurodegenerative diseases, Alzheimer's, Huntington, Parkinson, ALS, and so on. And for that, we use tiny worms. They're called Caenorhabditis elegans. These are nematodes, and they're 1 millimeter long. And these worms are transparent. They have 959 cells. So every adult worm has exactly 959 cells, no more, no less. And we know the location of these cells, and we express in these cells specific fluorescent proteins that can then sense the folding environment within specific tissues, in neurons, in muscle, and in the intestine. So if we shine a fluorescent light at these worms, and we could actually see how these misfolded proteins light up in different tissues of these worms. So now what we have done, we started feeding these C. elegans different bacteria from the human gut microbiome and asking question, what is happening to this protein folding across the different tissues in these worms? And what we notice is that when we feed specific bacteria to these worms, so when we colonize their gut with specific bacteria, now we see an induction or increase in protein damage. And that causes protein aggregation and toxicity that we see in patients with Alzheimer's, Huntington, Parkinson, and all of these other neurodegenerative diseases. So using that tiny nematode, we could actually study the influence that specific bacteria from our gut have on the brain health. And why is it important? It's important because we understand that somehow dysbiosis in our gut causes or is associated with the pathogenesis and the prevalence of these diseases. So we see very specific microbial changes, decrease in specific microbes, and an increase in another set of microbes in the gut of these patients with these neurodegenerative diseases. But we don't actually understand which bacteria are associated specifically with these conditions, or what do these bacteria do. So my lab is trying to first identify the bacteria, which bacteria specifically contribute to these diseases, and which bacteria can suppress these diseases. And we actually found both different types of bacteria because we screened all culturable isolates from the Human Microbiome Project and on the effect of each of these individual microbes on these protein folding sensors that we have in these tiny nematodes. So the idea of this part of the project is that we could identify specific bacteria and specific molecules that these bacteria could be producing that then contribute to disease pathogenesis.
Daniel, I think both of these areas are so cool, and I'm looking forward to talking in a little bit more detail throughout our conversation about some of these specific projects. And I know it's easy to be motivated by super exciting science, but I love getting quotes and sayings, mantras from everybody we have on the show to help motivate the rest of us out here who might be going through the slog of mundane things. So do you have a favorite quote or a saying or something that really motivates you in the work that you do or in life in general?
I actually have this quote underneath my email signature, and it reads, "Your work is going to fill a large part of your life, and the only way to be truly satisfied is to do what you believe is great work." And that's a quote by Steve Jobs. And I really like this quote because it gives me a motivation that keeps me going. It's the passion for science, for discovery, and for innovation that we could actually develop something. We could discover something in my lab that will contribute or affect the entire society. So I think that quote really gets me going every day, right? And even when I face a lot of failures, as we scientists do all the time, I know that at the end, this work is really important. So even if I do encounter these failures, I know that the work that we do is important, and whatever the results are, that will really keep me going and keep me satisfied.
Absolutely. I think kind of taking a step back and assessing the big picture can really help put things in perspective and help you really just appreciate that all of these efforts are going towards something really important. And you mentioned that Steve Jobs, who I know has inspired many people. Are there others that you look up to as maybe inspirational figures or role models or people who've just had a really big impact on you?
I was very lucky to have really good mentors, both as an undergraduate, as a graduate, and as a postdoc. But really the three people that contributed a lot to who I am today as a scientist is Rick Morimoto. He was my graduate advisor at Northwestern University, outstanding advisor. Sean Crossan and Howard Schuman were both of my postdoc advisors. But Sean Crossan really stood out. So he was a young faculty. He accelerated very fast, became a full professor within like 7 or 8 years. And he was always the to-go guy and the yes guy. So if you put a paper or a manuscript on his desk, it will not sit there. It will get published within a few weeks.
Oh, wow.
If you email him in the middle of the night, he will respond right away. If you ask him for a letter of recommendation, you're going to get that letter within a few hours.
Mm-hmm.
And he was so energetic. He had his own family and great career. And on top of it, he would run these long distance races. You know, he would go to like Arizona and run 50-miler. And then go to a conference straight and then fly back and come back right to a lab meeting. So very energetic with a lot of positive energy. And I think what I took from him is always grab that opportunity. I don't want to wait until tomorrow to do something. When somebody asks me to do something at work, I try to respond right away. And that sort of attitude that I got from Sean, what I learned from him during my postdoc.
Absolutely. It sounds like this was a tremendous influence on you. I have this mantra that I love myself that's sort of not later now. So get everything done now that you can, because you don't know what's going to happen in the future that might sidetrack you. So I love this story of this motivational and inspiring mentor that you had. And I'm curious to hear a little bit more about how you got on this path to begin with. So Daniel, can you walk us through your scientific journey, maybe starting all the way back at the beginning, and tell us what first sparked your interest in science?
I think the very early spark that I got, or the exposure to biology, was probably when my mom, I was little. I was probably around 7, 8 years old. We were living back in Poland. She got me this very well-built metal Russian microscope.
Ooh.
And Russians have really good optics. And this microscope, it was not a toy microscope. It was a very nice professional scope. So I really started using it to look at everything. And it was just blowing my mind. I was so fascinated with the micro world. And that sort of sparked the passion of curiosity and discovery. It's actually interesting because my passion for imaging and for microscopy followed me throughout my undergraduate and graduate and postdoc. And even right now, I have state-of-art microscope in my lab and we use in our work every day. So I think that was the first spark that really got me into the micro and the scientific world. And then we moved to the United States and I went to high school. And I really didn't want to spend too much time in high school because I wanted to do science and I wanted to go into a university. When I came to the US, I didn't know any English. I started as English as a second language, shelter classes, shelter biology, but I was able to graduate in 3 years. I learned the language within that time. And it's interesting because I still keep in touch with my first biology teacher, Ms. Donna Maybush, from my high school, and I give her updates from time to time. So then when I graduated high school, I went to the University of Illinois, Chicago to major in biochemistry. And that's when I was first exposed to real scientific research. So I started working in the lab immediately as a freshman, and I was doing research for the next 4 years. And I was just fascinated with it. I loved it. Initially, I was working in the biochemistry department. I was working with C. elegans already back then with Peter Okima. And then I started working in surgical oncology in med school, where I was working on combination of different phytoestrogen with chemotherapeutic agents and looking at the cytotoxicity in cancer cells. So I was doing that for 4 years. Great experience. And I always encourage my undergraduates right now that I mentor in my lab to really be exposed and be committed to research, because I know the importance of it early on will have a huge impact later on in their careers. So then when I graduated from UIC, I got accepted to Northwestern University, where I did my PhD studying the regulation of the heat shock response in C. elegans with Dr. Rick Morimoto. And then after that, I moved to a little bit south to the city, the University of Chicago. And I spent 6 years as a postdoc. And again, I was mentored by Howard Schuman and Sean Crossan. And that's when I was studying bacterial pathogenesis, specifically Brucella abortus. And I was developing these host-targeted approaches to target these intracellular bacterial infections. Then in 2018, I went on a job market, started applying. I got an offer at the University of Florida. And it was a great opportunity because I told you at the beginning, my family and I, we love traveling. So we've been to Florida every single year for the past 20 years. But we were never in Gainesville. And to be honest, I didn't really know where Gainesville even is on the map because we would always visit those coastal areas. But we came here for a visit and we loved it. It was a perfect environment. So for us, it was either we stay in that Midwest Chicago area, or if we move, we were really open to moving to Florida, pretty much only to Florida. So when this opportunity came up, we didn't think twice. We moved right away. And since then, I established my own lab where I do 50% research, 50% teaching. And it's been a really great adventure since the very beginning.
Absolutely. I think the winters in Gainesville are a little kinder than in Chicago.
Absolutely. Yes.
Well, it sounds like you've had a phenomenal journey. And I know you spent some time also working at the Argonne National Laboratory. Can you talk a little bit about what that experience was like and maybe how it was different from maybe a more traditional academic laboratory at one of the universities?
So actually, I spent a couple of years working at Argonne, and as a graduate student, I was a visiting researcher at Argonne National Lab, and I spent there over a year. It was a Midwest Center for Structural Genomics. So what I was doing, I was cloning different fragments of a heat shock factor 1, HSF1, and trying to develop monoclonal and polyclonal antibodies to different epitopes of HSF1. So it was a massive project because I had to clone something like over 20 fragments, purified all of them. And then I came back to the Chicago area and then started injecting both mice and rabbits to develop these antibodies. So I really got to know a lot of people at Argonne and at the Midwest Center for Structural Genomics. And I really liked that environment. So then when I was looking for a postdoc, I was also considering Argonne. And when the opportunity came up to do a postdoc in the Department of Microbiology, the University of Chicago, which is also associated with the Howard Taylor Ricketts lab, it's a regional biocontainment that is owned and operated by the Microbiology department at the University of Chicago. And that's where I spent about 3 years working in regional biocontainment, doing BSL-3 work. And I was working on a select agent. These are potentially weaponized biological agents. And that work was really fascinating. And I think that also goes along with my personality. I like to be more of a nature guy than a city guy. So Argonne is located in the middle of nowhere. And for me, being able to do experiments and then leave the lab and go out for a run in the forest was just amazing.
That sounds phenomenal. Was it nerve-wracking kind of working with all these quite high-level security measures and some of these potentially dangerous pathogens?
Imagine that you're holding a mouse doing an infection study, and if your needle slips and you poke yourself, you could potentially die from that infection. So it is definitely a research that is really high in adrenaline, I would say. But it was a really good experience. One of the funny stories is that when we were working with mice in the biocontainment, we would always have PAPR. This is like a literally HEPA filter that is sitting on a helmet on your head. So all the air that I would breathe was always purified. And I was working with a lot of mice. Everything was okay. But then when I moved all of my work back to Hyde Park to BSL-2, when we're only wearing a mask, not this whole HEPA filter, I realized that I'm allergic to mice.
Oh, really?
And I would get severe allergic reactions every time I would go into a mouse room. And I didn't know why. And it turned out that when I was at Argonne, I was not allergic because everything was filtered. Then when I moved to Hyde Park, I was wearing a face mask. That's when all the allergens was inhaled. So in Hyde Park, I was actually given one of these BSL-3 suits to work in the BSL-2 facility. So it was funny that whenever somebody would walk into a room, they would completely freak out because they thought I'm working on something very dangerous and deadly. So they would turn around and I just tell them, no.
It's me, not the work. Yeah.
I'm allergic. It's me.
Oh, well, that is amazing. It sounds like you had a fantastic experience in your training up to getting to where you are today at the University of Florida. And we mentioned some of the amazing projects that you're working on. Is there one in particular, Daniel, that is maybe your favorite at the moment or that has had some exciting developments that you would like to go into in a little bit more detail?
The problem with me is that I'm excited about everything that I do. And if I'm not excited about a project, I will not start it. And that's also the beauty of being a faculty, right? Of being a professor. You do experiments that you're really passionate about. But really, one thing that stands out is the concept of using bacteriophages, the concept of using viruses to specifically target and eliminate particular pathogens within the entire population of bacteria. So what we have done in the lab, we looked for these bacteriophages in wastewater treatment plant, in sewage water, and we also tapped into wastewater from the UF hospital to look for these phages. And bacteriophages, we'll find them anywhere where the bait bacteria, where the bacteria that they're attacking are present. So of course, they're going to be present in hospitals and they're going to be present in sewage. So we found about the 13 different bacteriophages that target specifically Pseudomonas aeruginosa. And these are bacteria that are ubiquitously present in the environment. They're opportunistic pathogens, and they mainly infect immunocompromised individuals, cystic fibrosis patients, or they love to infect skin burn infections, burn wounds. So what we started doing, we isolated these bacteriophages against these Pseudomonas aeruginosa strains. And right now we have a cocktail of 7 bacteriophages that can target over 90% of any clinical, environmental, or animal isolates of Pseudomonas aeruginosa. So it's amazing to even think about that we could use another biological system to specifically target and eliminate these bacteria. But what's really interesting is that it's not a novel concept, because bacteriophages were actually used as an antimicrobial treatment way before antibiotics were used. So for political reasons and from overabundance of antibiotics, the research into bacteriophage therapy was abundant after World War II. And Western medicine became more reliant on antibiotics, which at that point, during 50s, 60s, and 70s, they were overly abundant. There was the golden age of antibiotics, and we had antibiotics for every infection. Now the problem began to appear where these infections rapidly became resistant to almost every class of antibiotics. So right now we see bacterial infections that are pan-resistant. They're resistant to every single known antibiotic. So we're reaching for these old methods, to these non-traditional approaches that were first developed but were abundant over a period of decades. And these are really powerful therapies where a lot of studies are coming up right now where phage therapy is very effective and it has no side effects. So we could inject these bacteriophages into our bodies and they will specifically eliminate only the target bacteria. What I'm also very excited about is that this project overlaps with the other half of my lab that works on the identification of bacteria that contribute to neurodegenerative diseases. Because it turns out that one of the bacterial species that we found contributes to the disruption of that folding environment and induces that toxic protein aggregation is Pseudomonas aeruginosa. But it turns out that Pseudomonas aeruginosa actually colonizes about 8% of the general population, and that's asymptomatic colonization. But is it really asymptomatic? And what we showed last year is that these bacteria actually produce and secrete bacteria, and we call them bacteria-derived protein aggregates. So they secrete protein aggregates that then disrupt the folding environment in the host. So ideally, what we would want to do is to eliminate these bacteria if they indeed contribute to the pathogenesis of these neurodegenerative diseases. But how do we eliminate these pathogenic bacteria? We can't use antibiotics, because if we use antibiotics, what's going to happen? We're going to disrupt the protective commensal bacteria, and we're going to enrich for these gram-negative bacteria that are already intrinsically resistant to antibiotics. So we will enrich for the antibiotic-resistant bacteria and we'll destroy the protective bacteria in our gut. So we can't use antibiotics. So what do? We could use bacteriophages. So we actually already demonstrated a proof of concept where we could take bacteriophages in our C. elegans model, and we could specifically eliminate the bacteria that contribute to the disruption of the folding environment, and we could decrease the toxic protein aggregation. So I think this is going to be the future of approaching these complex diseases. And that's also a really nice crosstalk between antibiotic use, between antibiotic resistance, and the pathogenesis of these neurodegenerative diseases.
Well, this is a remarkable project, Daniel, and I love the idea of this ultra high precision treatment approach for some of these diseases. If this is found to be a direct link, what these bacteria are producing are causing these neurodegenerative diseases, I think the potential for the impacts are tremendous. But I know not every day in science is full of these exciting discoveries and promising results. There are a lot of failures, as you alluded to, and a lot of times things go wrong. So do you have a story of your own of a failure or a particularly challenging time that you had to endure and how you worked through it?
We are in the field where we are constantly failing. Our grants are rejected, papers are rejected, our experiments are not working. And that's something that I try to teach my students, that it's okay, right? It's not a failure. It's a learning process. But some of the epic failures that I had, well, one of them, what I already mentioned before, was that I spent about a little bit over a year at the Argonne National Lab purifying these heat shock factor protein fragments and developing antibodies. So at the end, after about a year of work, it turns out that none of the antibodies were able to detect the endogenous protein. I was able to detect the recombinant protein, but that was not the point. Nobody can ever generate antibodies against that specific transcription factor. And I tried for a year and also failed. I don't know if that was a complete failure because I learned a lot. And we also learned that it's really difficult to develop antibodies against that protein, or maybe almost impossible. But yeah, that was just part of a training. And then there are also personal failures as well, right? As we're not only in the lab, but even when we're scientists in training. So for example, when I was a postdoc, and that story might be relevant to other postdocs or help them go through difficult times. But when I was a postdoc, I had 3 children. So that's already difficult. Being a postdoc working full-time, 3 children, my wife was not working, and being on a postdoc salary with a family of 5, that's challenging. And at the time, my dad passed away and he was financially helping me out. So this whole period of my life became like really difficult. But I knew I had to endure. I have to survive the tough times because I really wanted to be a faculty, really wanted to have my own lab. And I spent 6 years as a postdoc. And that's a big amount of time. But you just push through the tough times and endure and make it work. And I'm a religious person, so I always put my faith in God that he will guide me through the difficult times, and things just went the right way.
I'm so glad you shared this story because I think a lot of people think about the challenges, the failures, just kind of from this scientific perspective. But becoming a scientist is a personal investment. There are a lot of personal challenges and struggles along the way. And I think you haven't failed until you give up. Like you said, you just keep going if that is your dream, if that's something that you're really passionate about. So I'm glad to hear this story had a happy ending for you. And I really appreciate you kind of giving us insight into what this process was like for you, this struggle developing the antibodies, and also just this difficult time in your career and in your life that you had to work your way through to get to where you are today. And I think you've had a lot of successes to celebrate, Daniel. So we don't just want to focus on the tough times, the failures. So do you have a favorite success story, whether this was a big win or just a small but particularly meaningful victory for you?
So I think I would have to ask myself, right, how do I measure my success? And in academia, it's really funding and publications, right? That's a really great success. And of course, the students that we train. So every publication that we publish, we celebrate. Every grant we get, we know that we're secured for the next few years of innovation. I just recently graduated my first PhD student, Dr. Alyssa Walker. And that was amazing because it took 4 years. And 4 years, that's pretty quick for a PhD. But Alyssa was able to publish 4 first authorship papers within 4 years. And I think that's a really great success. And it underlines the success of the lab, of the project, of the funding opportunities that we got. And of course, the novelty of this project. So Alyssa was working on the project where we're studying the effect of the gut microbiome on these neurodegenerative diseases using C. elegans as a model organism. So it's a relatively new area of research. And it's definitely exciting because now we're starting to approach our understanding of these diseases from a very different perspective that we never thought or considered before. And that's the microbial contribution. Not only what our bodies contribute to these diseases, but microbes are actually factories of molecules. And how do these molecules affect our body? We know they do. We don't know how, but understanding that, it's one of our goals. And I think Alyssa, her success as a graduate student and being able to finish so quickly, and she's continuing working in my lab as a postdoc right now. but I think that really emphasizes one of the success stories that we have. And there are also other publications. Recently, we just published a very interesting article in Frontiers in Microbiology, where it's an industry collaboration with natural immunogenics, where we are studying the effect of these silver nanoparticles in combination with antibiotics. So what we found is that if we combine these silver nanoparticle spheres that are about 3 nanometers in diameter, we could potentiate the effect of aminoglycosides by over 20-fold. Now, why is this important? Because aminoglycosides are very toxic antibiotics, and they're routinely used in pediatric medicine. And one of the side effects is irreversible loss of hearing. So what we now have is the opportunity to lower the concentration of these antibiotics to non-toxic concentration, while at the same time keeping the efficacy the same or even enhancing the efficacy that can work against antibiotic-resistant strains. So I think this is a very fascinating approach and a really fascinating discovery, because it provides another approach in which we can resurrect antibiotics that are not only expiring, but are so toxic to the patient.
Well, congratulations to you and your lab members, and particularly to Dr. Alyssa Walker, your first graduated PhD student. And I know breaking into these really new cutting edge areas can be tough. Was this something that it was a struggle to get funding for some of these new projects because people weren't so sure about kind of the rationale or the concepts behind them?
Well, as a new faculty, I had my startup, so I had more of a wiggle room or where I should invest my time and my startup into which project. So I started a couple of projects. Actually, I'm following up on all of the projects that I started, and I was able to get funding for all of these projects and keep them going. But funding just comes. I think when I stay concentrated and I focus on a project and I'm really passionate about it, I constantly seek out for available funding, and there's always going to be funding, right? We just have to find it, but it's out there. It's definitely out there. So this funding for the silver nanoparticle and aminoglycoside project, that came from our industry partnership, and it was great. The university had a very specific funding program where the university matches industry collaborators with academia collaborators and provide matching funding. So I got funding from both UF and our industry partner. So I think these type of funding mechanisms are not well explored, but they're available and they're out there, and they can result in very fruitful collaborations.
Absolutely. And I love that you are open-minded to considering these alternative sources of fundings that many people don't even think about when they're putting their traditional NIH or NSF grant packages together. So I think being creative and open-minded can definitely be helpful in science. And we've talked a lot about the work that you're doing in the lab and some of the things that you like to do for fun outside of science. And we are always encouraging our listeners out there to pick up a book and try to read for fun to expand their horizons a bit. So, Daniel, do you have a favorite book that you can recommend for me and our listeners, whether it's related to science or not?
I think I would recommend the last book that I just recently finished reading. It's The 5 a.m. Club by Robin Sharma. And what's really fascinating about this book, and I would recommend it to every graduate student, especially because it really tells you how to well organize your life, but still be productive and keep that work-life balance. And it's kind of funny because when I was reading it, some of the things that were emphasized by this book, I was already doing it without even realizing. I don't know if it's just such a busy lifestyle that we have to crank all the productivity in a shorter period of time and still keep that life-work balance to keep sane. But that's definitely one of the books that I would recommend for not only PhD students, I recommend it to everyone who wants to be productive. I actually bought it for one of my students in the lab a couple of weeks ago. Left him a little gift surprise on his desk.
I love it. I'm an early riser myself, so I subscribe to this. Kind of getting up and moving in the morning helps me just be more productive throughout the day. Otherwise, if you have kind of a slow start, it turns into a lazy day and not much gets done.
Absolutely.
Well, great recommendation. And we've talked about some of the different opportunities you've had as a scientist. You've been able to travel a bit for your work. You've worked with different collaborators. You've gone to conferences. Do you have a favorite place, I know, that your science has taken you? And if so, where?
I think traveling is one of the perks of being a scientist because you get to network with amazing scientists and you get to see places where normally you wouldn't go. I think one notable place that I went to is Suzhou, China. And I was a graduate student and I was invited to give a talk at one of the meetings. There's a Cold Spring Harbor replica of our Cold Spring Harbor laboratories in New York. There's another one in Suzhou, China. So I flew into Shanghai, and it turns out that two of my best friends were living in Shanghai. They were from Chicago, but they were living in Shanghai at that time. So I stayed with them for a few days. Then I went for a week to the conference in Suzhou, which is just about 100 kilometers west of Shanghai. And then after the conference, I went back to Shanghai, and then my brother flew into Shanghai from Chicago. And we were just traveling all around China for the next week. Science also gives us the opportunity to explore different places. And as a graduate student, I think it gives you the opportunity to see things that normally you wouldn't be able to afford to visit. So that was definitely one of my notable experiences. One interesting thing about this trip is that it happened during the Chinese national holiday. And that's when the entire China has a whole week off. So there are millions of people traveling, and I didn't realize that, but it was packed. And we went to every major city in China, and these cities were just packed with people. And the communication was also a little bit difficult to come through, but we survived. It was definitely an interesting experience and adventure that stemmed from the regular conference.
Oh, this sounds like a phenomenal experience, and to be able to explore so many different cities in, like you said, a very far away place that you may not otherwise get to go. And you also alluded to just some of the amazing people that you get to network with as a scientist. And I think the people in science are sort of like the undiscovered gem, because there's these stereotypes out there about what scientists are like, and they're often portrayed as being very serious, very critical, and working by themselves in the lab. But that's not the case in so many situations. I think some of the funniest and most creative people I know are scientists, and there are quirky traditions that spring up and just hilarious memories and moments that you share together. So, Daniel, do you have one of these stories of this human side of science that goes against the stereotypes that you can share with us today?
So, for example, in my lab, we try to be social. So when a graduate lab that I came from at Northwestern, from the Morimoto lab, we were very social. It was like another family. And it's funny because one of my best friend, graduate student who was my colleague, she was sitting right next to me, Caterina Silva. We were joking that she is my lab wife, right? So I have a wife at home, and then she is my lab wife because we spent so much time together, working together. We had a bench next to each other. So I think what I learned from my graduate experience is that social component is so extremely important just to stay sane and to keep the right work-life balance when we are most of the time locked in the lab and we're performing experiments without really talking to anyone. So it's a little bit, I don't want to say that scientists are antisocial, but it's not unusual for me when I was a postdoc or graduate student to be locked in a microscope room for hours and just not talk to anyone. I'm just looking at my cells. And it's funny because like my brother would ask me, hey, did you talk to your worms today? Or my wife would ask me, hey, how is your cell culture doing? You know,
your friends at work.
Yeah. They make these small jokes that I had no friends, but my friends are my biological specimens that I work with in the lab. So I think that having that really social interaction and social bond with the lab that you were working with, that really helped me when I was a graduate student. And then in my lab right now, I also encourage different activities. So last year, I took the whole lab. We went to Cedar Key, which is a town just 45 minutes west of Gainesville. We rented kayaks and we were kayaking on the Gulf of Mexico between different islands. And that was a great bonding experience between students. And it's a great way to get out of the lab and actually do something else, but still do it as a team. And I think that that type of events, I always encourage them and I support these type of adventurous outings. We also have regular barbecue dinners at my house. We always have one for the holiday season, for Christmas season, before New Year's. And we have another one at the end of the school year to celebrate the end of the school year. So I think these social events bring the lab together and they help with the productivity because the environment in the lab is more social as well.
Absolutely. I think developing this sense of community is so important in science. And you sort of, when you have that camaraderie, it's easier to bounce ideas and questions off of each other. And I know oftentimes science does involve a lot of solitary work. So just kind of having this background of a lab dynamic, a group that you can count on to be there for you, I think is so important. And we talked about some of the big questions you're answering in your lab. And I know oftentimes things like funding, technology, time, staff, feasibility, whatever the case may be that's holding you back, keeps you from answering the questions you want to answer. But Daniel, if we took away these barriers and gave you all of the resources you could possibly dream up, is there one particular question that you would want to answer first?
There's so many questions. I think the two problems that my lab is working on, antimicrobial resistance and neurodegenerative diseases, are both so important right now. There is absolutely no cure for them or even effective treatment. And we could treat cancer, we could cure cancer, we could treat diabetes, heart diseases, right? There's nothing for neurodegenerative diseases. So I think if any part of my research can contribute to understanding the microbial contribution to these diseases and actually demonstrating that these microbes do play an important factor in the pathogenesis, I think that would be my biggest win if we could demonstrate that.
Absolutely. I think that is a worthy way to dedicate these unlimited resources. These are enormous questions that you're working to answer in the lab, and the neurodegenerative disease one is near and dear to my heart and my research interests as well. And I know we've shared a lot with our listeners today, but I'd love to end with advice. So, Daniel, do you have a piece of advice that somebody gave you at some point in your life or your career that really helped you that you can share with our listeners today?
I don't know if this is anyone's particular advice, but it might be more of a mindset not to think about failure as an actual failure as a person, but think about it as a learning process to get better. Again, this is something that I had to relearn as both graduate student, as a postdoc, and even as a faculty, because we want to succeed in everything that we do. But we're in the field, like I mentioned before, that where we encounter a lot of failures, but they're not failures. It's a process of how we get better. If we, for example, don't get a grant, we didn't fail at getting that grant, right? We resubmit, we make it better, we improve it. Same with papers. We addressed reviewers' comments, we resubmit, it gets accepted. So I think the advice would be to think about failure as a learning process to improve in what we're doing.
I love that advice. And I think seeing these, maybe we'll call them setbacks, not as the end point, but sort of just as part of the process, part of the journey that will end in a success, I think is a really good shift in mindset for people, particularly during some of these difficult training times, like our listeners might be going through their PhD or postdoc, things like that. So wonderful advice to share. Is there any other last piece of advice, Daniel, or a last note of inspiration that you would like to end our conversation on today?
Never give up. Keep going. Just like I had a hard time when I was a postdoc, I kept going. One of my favorite stories is about Linus Pauling, and he published a structure of a DNA even before the actual structure was known. And that was a model. And he was a great scientist, but he published the triple helix with phosphate molecules on the inside of the helix. Now, based on the elementary chemistry, we know that phosphates have negative charges and negative charges repel each other. So chemically and physically, it's impossible to have these phosphates facing each other on the inside of the triple helix. So it was a very elemental mistake. And he was really set back by that publication, but he never gave up. He didn't give up. He kept going, and he got 2 Nobel Prizes after that. So it's not really a failure. Might have been a mistake, but you keep going. And when you keep going and you're persistent about your passion, about the direction of your research, then eventually you will get there. You're going to be successful.
Definitely. I think that is such a wonderful message to share, and comforting to sort of reflect on the fact that even Nobel Prize winning scientists, people like Linus Pauling, have made some of these sort of seemingly silly mistakes looking in hindsight. So great message to end our conversation on today. Daniel, if our listeners would like to learn more about you and more about what you're doing, what is the best way for them to do so?
I'm pretty active on Twitter. My Twitter is at 360science. Very simple. 360science. I was able to get that domain a long time ago.
I'm impressed.
And usually I share news updates about my research, about my teaching. I'm actively involved in teaching. I'm also actively involved with the National Institute of Antimicrobial Resistance Research and Education, NIAMRE for short. I'm organizing their annual conference. I'm going to be hosting it at UF this coming May. And yeah, I do a lot of updates on Twitter. I think that's the best way to get in touch with me. There's also on Twitter, you can find my lab website. And that's also how I stay in touch with my students.
Fantastic. Well, listeners, definitely get connected with Daniel on Twitter. And Daniel, it's been such a pleasure to chat with you today. Thank you so much for your time.
Thank you very much, Marie, for having me here.
Well, it's been great to chat with you, and listeners, wonderful to have you here with us as well. We hope you'll join us next time for another episode of People Behind the Science.